Influenza, or the flu, is caused by one of several types of viruses that can spread quickly from person to person via airborne particles. If the particles are inhaled, they will travel down the trachea into smaller and smaller airways. During an influenza virus life cycle, viral surface proteins bind to receptors on the surface of cells that line the respiratory tract. The virus is then engulfed by the cell into a compartment or endosome. Due to changes in the pH environment within the endosome, the hemagglutinin HA protein on the surface of the virus undergoes a conformational change. This exposes the buried fusion peptides, allowing them to insert into the endosomal membrane. Both membranes fuse, which creates an opening for viral RNA to exit the endosome and enter the cell nucleus. The viral RNA is replicated within the nucleus. Finally, the RNA copies are joined with the newly formed viruses as they bud off the surface of the cells. The therapeutic antibodies can bind to the stem of the HA protein. The viruses still bind and are engulfed by the cells. However, these novel antibodies prevent the conformational change needed for the virus to fuse with the endosomal membrane and release its viral RNA. This stops the virus life cycle and the virus is degraded.